All right. Hi, everyone. My name is Amy Klippinger, and I am the Associate Director of PETA's International Science Consortium, and will be co-moderating today's webinar with Dr. Warren Casey from the U.S. NTP Interagency Center for the Validation of Alternative Toxicological Methods, also known as NICETM. I'd like to thank you all for joining the first webinar in our 6-part series. The goal of these webinars is to address current practices for acute inhalation toxicity testing, looking towards the development and implementation of alternative approaches that reduce and replace acute inhalation testing in animals for both global regulatory agencies and non-regulatory purposes. The slides from today's webinar and a recording will be posted within a couple days at the link on the screen. You can look for the automated email from GoToWebinar after today's presentation, and that email will contain this link, so you don't need to jot it down right now. Also, after today's webinar, there should be a feedback form that pops up in your internet browser. Please do fill that in if you have time, as it will help us to make sure we address issues that are important to you in this series. Everyone is muted now. If you have a technical question for me anytime during the presentations, you can use the chat box, which should be located in the panel on the right-hand side of your screen. If you have a question for one of the speakers, we will hold all the questions until after both talks. To ask a question, please type it into the chat box, which is part of the toolbar on the right side of your screen. Warren will ask your questions from the chat box to the speakers during the Q&A period, so please send them in anytime during the webinar. I also want to point out, importantly, that this webinar series is being organized with input from multiple organizations, and I'd like to thank each of them. You can see the complete list of these organizations at the same link that is on the screen now. Lastly, before I introduce our first speaker, I want to mention the other webinars that are coming up in this series. The next webinar on in vitro methods and dosimetry will take place on April 26th, and the third webinar on in silico methods will take place on May 26th. You can find more details about these and the other upcoming webinars online. Also to note, if you're registered for this webinar, you will automatically be registered for all 6 webinars in the series. This means that you'll get a reminder from GoToWebinar for each webinar. However, there's a lack of synchronization between GoToWebinar and Outlook, so the later webinars are not automatically added to your Outlook calendar. So you'll have to manually add the appointment reminders to your Outlook calendar if you would like them there. Okay. Speaking first today will be Ian Indans, who is a regulatory toxicologist working for the UK Health and Safety Executive's Chemicals Regulation Directorate. He represents the UK at REACH test method meetings and ECHA's Member States Committee as an expert toxicology advisor. He has a long-standing interest in the application of 3 R's in regulatory toxicology, in particular in vitro approaches to skin and eye irritation testing, refined approaches to acute toxicity, and use of the extended 1-gen reproductive toxicity study for REACH. His talk today will focus on the 3 R's, current needs, and future prospects for acute inhalation toxicity testing. And with that, Ian, you can go ahead.
Yeah, thank you, Amy, for that kind introduction. Thank you for inviting me to give this talk this afternoon, although given the number of people we have online, I suspect there are different time zones. Either good afternoon, good morning, or good evening. And as said, my talk is on acute inhalation toxicity testing, where we are right now with 3 R's, what we need from inhalation tests at the moment, and a quick look at what our future prospects may be for minimizing acute inhalation toxicity testing. My name's Ian Indans. I'm a regulatory toxicologist. I work for HSE. Chemicals Regulations Directorate is one part of a sort of larger organization. and just give you a quick flavour of what we do at HSE in CRD. We're the competent authority for REACH, which is the Industrial Chemicals Regulatory Scheme. We're the competent authority for the EU Biocidal Products Regulations, competent authority for Plant Protection Products Regulations. We're also involved in CLP regulations and also OEL setting. And just to, for those of you who don't necessarily know much about the geography of the UK, we're based in Bootle in Liverpool, and just to try and orientate you, we have two famous football teams here, one being Liverpool, and I couldn't possibly go on without mentioning the other side of the divide, Everton FC, that's the blue half. And for those of you who are not necessarily into football, then there's a sort of a famous brand which came from Liverpool, which is the Beatles in the bottom corner. So enough of sort of where we are and what we do. So my talk this afternoon is on acute inhalation tox testing, 3 R's, current needs and future prospects. What I hope to do this afternoon is look at what we as regulators use acute inhalation toxicity testing information for, what the scope is for the use of the 3 R's for minimising future testing, that is reduction in animal use, refinement of acute inhalation toxicity testing procedures, and perhaps more long-term is the prospect of replacing current testing with alternatives such as in vitro alternatives, computer testing, that sort of thing. And perhaps finally look at some of the obstacles that maybe get in the way of minimising inhalation tox testing. So the three schemes I'm going to look at first this afternoon are REACH, the Industrial Chemicals Regulation, and the Biocidal Products Regulation and the Plant Protection Products Regulations. All of these are from an EU perspective because that's the only way I can give you a perspective of inhalation tox testing. Because these are all regulations, then they're all direct acting, which means that across the European Union should be a level playing field in terms of testing requirements. And just to give you a quick overview of what the aims of REACH are, pretty similar for the plant protection products and biocidal products regulations, the aim of all of them is to ensure a high level of protection of human health and the environment. And this is actually quite critical, because if you're thinking of alternative tests, what we should get from alternative tests is no loss of protection. The free circulation internal market is more a European Union idea, but it certainly translates across more worldwide, because what we shouldn't see is toxicity testing being used as to provide a barrier to trade. So developing an alternative test and free circulation of goods and services is important. And lastly, with REACH, one of its aims is to develop and promote alternatives to current testing. And now looking more closely at what REACH asks for in terms of acute inhalation toxicity tests. REACH doesn't ask for tests per se, it asks for information, which means that could be provided through a standard test, or it could be provided through something else. And standard information requirements in REACH, they are tiered dependent upon tonnage. There are four tiers. Annex 7 and 8, as you can see on the slide in front of you, 1 to 10 tons per annum. Annex 8, 10 to 100, 9, 100 to 1,000, and Annex 10 is 1,000 tons per annum plus. Looking back at Annex 7, there's no requirement for an acute inhalation toxicity test. The only real in vivo test is a requirement for an acute oral study. As you move up the tonnage to 100, or 10 to 100 tons per annum, there's a requirement for a second acute in vivo study. That could be inhalation or it could be dermal. And in column 2 of X8, you'll see the requirements for an acute inhalation study. Essentially, it's if there's some exposure by the inhalation route, then one must consider the need for inhalation testing. But in contrast to Annexes 9 and 10, Annex 7 and 8 don't require a testing proposal. Those of you not familiar with REACH, at higher level, Annex 9 and 10, the high-level test, they must be accompanied by a testing proposal, and only when permission has been sought through ECHA's Member State Committee and that has been confirmed can a high-level test be carried out. But Annexes 7 and 8, it's at the registrant's discretion what testing in terms of inhalation or dermal that they do. REACH, as one of its main aims, is to minimise animal testing, and it says in Article 13 that new animal testing should be taken as a result. And as you've seen previously, there's essentially a specific waiver in Annex 8, 8.5.2, only inhalation test if there is significant exposure by the inhalation route. REACH also includes in Annex 11 a number of general adaptations from the standard information requirements. They start out use of existing data, so that's not data which is generated by a standard OECD test guideline, but it might be one which is generated which is an equivalent to an OECD test guideline. It permits the use of tests which may have been done for the Japanese or the US jurisdictions some while ago and aren't quite the same as modern guidelines, but give you roughly enough to work with. Weight of evidence is slightly different. That pulls together individual fragments which individually don't give you enough to cover a REACH standard information requirement, but taken for inhalation toxicity testing, for acute inhalation testing, it's unlikely that this will be very relevant. It's more relevant for repeated exposure. REACH also foresees the use of QSAR's structure activity relationships for prediction of toxicity endpoints, but if those are used, that when registrants are submitting the data, they must submit them using the standard OECD formats, the QMRF, QSAR model reporting format, QPRF, QSAR prediction reporting format. That's what is expected if you use those types of data. REACH also permits the use of in vitro data, either validated to OECD test guidelines or those are in the process of being adopted. That's set out in Annex 1.4, and that's certainly something possible for the future. One of the more current methods for REACH waivers is grouping and read across. I'm saying, can we pull a group of chemical substances together and read across from some of them to predict toxicity outcome of others? It's certainly possible with acute inhalation testing. So that's something that would be powerful for minimizing animal testing. And lastly is the feasibility or not of actually testing material. If something's explosive, flammable, then it's not desirable to actually do an inhalation toxicity test for good reasons. Those are the sort of things in the general waivers and can be used by registrants to minimize inhalation testing. Looking slightly more broadly at what the plant protection products and biocidal protection products regulations require in terms of active substance data requirements, in contrast to REACH, the information requirements are tonnage independent. So if it makes 1 to 10 tons of a biocidal active substance, the requirement is for all the data requirements from acute studies all the way through to carcinogenicity studies. In terms of inhalation, there is a requirement to do an inhalation study if it has a particularly significant vapor pressure, if the substance is a powdered material and it's inhalable, or I say significant inhalable fraction, or the active substance is included in products that's sprayed. This is common to both biocides and pesticides. And what we do sometimes see, which for biocides and pesticides, we do sometimes see active substances being milled and then inhalation tested. This is not a requirement for biocides and pesticides in the European Union, but we do sometimes see it for non-European regulatory schemes. And this can cause problems certainly for classification and labelling, and perhaps something that we strongly hope not to see in the future. The plant protection products, you're also required to produce an inhalation study on the product if the substance is, or if the product is going to be used as a gas or a liquefied gas, smoke generating, etc. The key is that there's going to be some inhalation exposure, and the same needs apply in terms of particle size and vapor pressure. And critically, what do we do with the data for both biocides and pesticides, and for REACH? We have to do something with it. We classify and label the material in terms of UNGHS, or in Europe we use the translation of UNGHS, that's in the classification and labeling products regulation. We classify for acute toxicity, that's simply acute lethality. We identify, or try and identify, critical target organs, and if we can identify some of those, classification under STOT may be required, specific target organ toxicity, following single exposure. And something that's particularly useful that we may get from acute inhalation testing is whether the substance is a respiratory tract irritant or not. Normally the key data there is from humans, but we very rarely get that, and so good histopath from a standard acute inhalation tox test is actually very helpful here. We also may well do risk characterization, that in terms of biocides and pesticides it may be acute reference values, but we very rarely do risk characterization for workers, long exposure for consumers from an acute study, because that only tells us about single exposure, and generally there are a few things which are people are only exposed to once. It's more repeated or discontinuous repeated, so acute tox data less useful there, but they can be useful for approval of biocidal products and pesticides. And one of the things which they are commonly used for is land use planning. In terms of hazardous installations, they can be used to inform on when things like schools, hospitals are being built near a hazardous installation, how far away they should be built in order that there's unlikely to be a risk should there be an industrial incident. But traditionally, historically, the data that we've generated for land use planning, classification, labelling have been based on point estimates of the LCD that come from the 403 studies. And we're going to look very quickly at how the 3Rs might be applied to acute inhalation testing. That is refinement, reduction and replacement of animals in inhalation testing. The historic traditional way of generating data for acute inhalation testing has been using Test Guideline 403. That's been with us now for a good number of years. Can use up to 40 animals per substance and generally uses lethality as the endpoint. More recently, we've developed OECD Test Guideline 436, which certainly offered significant benefits in terms of animal numbers, probably reducing by about a quarter compared to 403, but 436 still retains death as its endpoint. And very recently, the UK has been involved in developing draft test guideline 433, the fixed concentration procedure. It's comparable to 436 in a number of ways, but instead of using death as an endpoint, it offers a predictor, something called evident toxicity, as the endpoint, and the intention is that you don't go to the next highest concentration which would cause severe toxicity or lethality. Interestingly, both 433 and 436 have moved away from using a point estimate of the lethality and use fixed concentrations instead. Just want to give a few bits of information on 433, the fixed concentration procedure. It uses evident toxicity, moving away from death as an endpoint, and it is the inhalation equivalent to the fixed procedure. A few of you may well be familiar with that. We've had 420 with us now since 2000, 2001, and there should be a lot of experience of using evident toxicity rather than lethality as an endpoint. We've defined evident toxicity for the fixed concentration procedure as clear signs that predict exposure to a higher concentration will cause severe toxicity or death in most of the animals that are being investigated. What evident toxicity isn't? It isn't the severe signs themselves, it's a predictor that they would occur if the next highest concentration were to be used. And what we say with the FCP is we stop below that concentration. And the intention of the FCP is that testing at higher lethal or severely toxic concentrations can be avoided. Looking further ahead now, can we actually see a time when we could replace acute inhalation tox testing completely? That there is almost certainly going to be a requirement for the foreseeable future for information from acute inhalation toxicity tests. We see with the biocides and pesticides that there are standard data requirements. For each there's at least a need to consider inhalation testing. So I think for the foreseeable future there will be a need for inhalation testing. And unlike things like skin irritation testing, eye irritation testing, and even skin sensitization, it's unlikely that the standard models will be replaced anytime soon. Although I think now maybe the world's changed ever so slightly. With the advent of the organ on the chip by the Wyss Institute, it may well actually give us the possibility of developing, if not a complete replacement, a very partial replacement. It's perhaps worthwhile thinking about what the replacements would have to give us. They'd have to give us information which could be useful for regulatory purposes, but it doesn't necessarily have to give us LC50 values. For the organ on a chip, it may give us information on histopathology, on target organ toxicity, those sort of things. And it perhaps partly is us as regulators to develop ways of using these new data rather than keep on saying they don't give us LC50 values. Lastly, I'd like to look at perhaps some of the impediments to further reduction in acute toxicity testing. So the biggest hurdle that we faced with 433 is the acceptance of evident toxicity across other regulatory jurisdictions. It certainly poses some challenges to current regulatory needs. And it might be that what is required there are new regulatory approaches which don't need a point estimate to the LC50. So if you're doing land use planning into the future, can we do it with a range estimate rather than a point estimate? I mean, it's possible that the OECD mutual acceptance data provisions will help, but they will help so far because they do say that a test that's done in one regulatory jurisdiction should be acceptable in another, which is fine. But if the regulatory requirements in that jurisdiction require point estimates of the LC50, then although we've developed new refined models, the chance of them used are not likely to be very great. So perhaps what's actually needed in parallel to developments of new test methods is to look at whether we can revise national information requirements both within the OECD member states and non-OECD member states. For instance, the bigger emerging markets such as China, Brazil and India. I think it's critical for new test method development that we try and achieve this, but it will only be achieved through dialogue. I think there's no point in us in Europe pursuing very refined test methods if no one else is going to take them up. Equally, there's no point in us in Europe standing and shouting and saying, we know what's best, you must follow us. I think we have to bring everyone along with us, not just regulators. I think this poses challenges to the political community too. I think with that, I'd like to bring my talk to a close and happy to take any questions at the end of the presentation. Thank you.
Thank you, Ian. So we will hold all questions until after the second talk, but please do send in any questions now using the chat box. Speaking next will be Dr. John Hotchkiss, who is the Senior Inhalation Toxicologist and Technical Leader of the Respiratory Toxicology Laboratory in the Dow Chemical Company's Toxicology and Environmental Research and Consulting Laboratory in Midland, Michigan. John has more than 30 years of experience in respiratory cell biology and inhalation toxicology, with experience at Lovelace Respiratory Research Institute and Michigan State University before starting at Dow about 15 years ago. He has conducted acute, subchronic, and chronic guideline and mode of action studies, as well as in vitro studies for environmental pollutants, tobacco smoke, and industrial chemicals.
Well, thank you very much for that introduction, Amy, and thank you for inviting me to talk today. What I'd like to talk about today, and Ian's introduction is really a good foil for my presentation, I believe, is I'd like to focus more on the nuts and bolts of acute inhalation toxicity testing and see where there is potential for alternative methods to fit into the scheme. First, as an outline, I'm going to talk about why we're testing, what the current testing guidelines are, a more detailed analysis of how we test, the potential alternatives that are available, and opportunities and challenges as we move forward. First, why in the world do we do acute inhalation tox testing? Well, first of all, it's a major route of human exposure in the environment. The respiratory tract provides a really unique interface between the environment and the systemic circulation that's unlike other organ systems. We have both an upper and a lower respiratory tract, which are distinctly different morphologically, and yet both are important in terms of how inhaled materials are processed and absorbed into the circulation. We test in order to understand exposure response data for hazard identification, and it's through the in vivo exposure mode of testing that we really get a full integration of test material properties, the deposition of the test material, its absorption, transport, metabolism, and elimination within the organism. And then finally, it's really important to identify the critical responses to inhaled materials, because the respiratory tract both is a point of absorption, but it's also potentially a target organ. So we have to differentiate between port of entry effects, where the cells and tissues of the upper and lower respiratory tract are the targets, versus systemic effects, where there are more generalized effects on other internal organs and tissues. So how do we use the data that we generate? We use it in hazard identification to assist in guideline testing for registration, classification and labeling, handling and shipping of the materials, and it really forms the core of the safety data sheet information which is associated with any product on the market. We also use it for risk assessment, primarily for setting occupational exposure levels for short-term exposures, and it's also critical for emergency response regulations and exposure levels. And so this is very important for first responders in terms of the potential toxicity that they may experience in the case of unexpected release or an accident. And then finally, there's product stewardship, where we can use this data to help fill data gaps, include it as part of a read-across strategy, develop new tests to understand the impact of reformulation of pre-existing products, and then also to use it to help in new product selection during the development cycle. So let's step back and see what the goal is. So we have to always remember that no matter no matter the test that we're conducting, the primary goal is to understand the hazard properties of an inhaled test material as it relates to humans. Now, our current testing paradigm uses in vivo exposures, principally of rodents, as predictors of human responses to these same test materials. But what we would like to do as we move forward is to introduce the idea of perhaps using in silico predictive methods, non-animal methods, or in vitro methods in order to predict responses in humans. Now, while this slide really depicts sort of the direction that my laboratory is headed. This slide really sets up the regulatory landscape in which I live in. And so this is reality versus where we want to be. So this slide provides a summary of the current and proposed US EPA and OECD acute inhalation toxicity guidelines. So across the top, I've got listed the EPA 1300 series acute study, and then OECD test guideline 403, the 436, and the proposed 433. So typically the exposure duration is 4 hours, although for certain types of regulatory considerations, very short 1 hour exposures are often preferred. But in general, the current paradigm is to use a single 4 hour exposure and then to extrapolate to shorter exposure times. The preferred exposure method is nose only, although in the case of gases, you can use whole body exposure. In the case of, and then 4 hour exposure then is followed by a 14-day post-exposure recovery period, during which times clinical observations, body weights, time to death, and other observations during exposure, and then gross necropsy at the end of the sacrifice are taken into consideration. For aerosols, the mass median aerodynamic diameter, that's the aerodynamic diameter of the particles that the animals are exposed to, have to fall within a range of 1 to 4 microns. And that wasn't just made up. That range of particles is based on the assumption that we're testing in rodents, which are obligate nose breathers. And that particle size distribution assures deposition in all regions of the respiratory tract without predisposing one site or another for the majority of the test material. Now, EPA 1300 and 403 are really quite similar to each other. They both provide a point estimate of the LC50, but in fact, they do differ. For instance, the OECD guidelines differentiate between exposures to gases, vapors, and aerosols. Now, this difference then is reflected both in the limit concentration for the different categories of assessments, but it's also reflected in the concentration bands or ranges that are associated with the hazard categories that I'll show you in just a moment. 403 also has a secondary protocol, a C times T protocol, which can be used when needed. This data is more amenable to the use in probit analysis to define exposure response curves, and it provides a better estimate of toxicity at the lower concentration boundaries of exposure. Now, 436 and 433 are both refinements of the traditional 1300 and 403 methods, which can reduce the number of animals used. And unlike 403 and 1300, they provide not point estimates, but rather toxicity range estimates based on stepped exposures at fixed concentrations. And as Ian pointed out, 403 introduces the concept of evident toxicity as an alternative to lethality as an endpoint in these acute studies. Now, summarized on this slide are the acute inhalation hazard categories based on the globally harmonized standards. And listed here, I have categories 1 through 5 for GSH. EPA does not use GSH categories nor warning phrases, nor and the hazard categories are based solely on test material mass concentrations, as opposed to those outlined by GSH. And so what I have depicted here are both the categories from most toxic to least toxic, and the associated pictograms and warning phrases that are associated. And then within each category then are the concentration ranges which correspond with the toxicity categories for testing. So I've given you an idea of what data the current acute inhalation guidelines generate. But here's what's missed. First of all, there's no histopathology of the respiratory tract or other systemic organ systems. The types and severity of exposure-related lesions are not reported. Any persistence or reversibility of the lesions, therefore, are unknown. And information on target organ toxicity is not available. And there's no data related to mechanism of toxicity. Now, the guidelines all say that you can add to the core group of information that's outlined in the guidance documents. However, and in fact, we have protocols which are enhanced acute inhalation protocols that we use principally for internal decision making, because unfortunately we found that depending on the jurisdiction that the data is supplied to, there can be a misunderstanding of how to use the data. And so especially for non-toxic test materials, we find that often the histopathology data or reversibility of the lesions or the description of the lesions is often misinterpreted and adversely affects the hazard category assigned to the test material, independent of the key issue of lethality that the acute inhalation toxicity protocol is designed to assess. So somewhat ironically, the guideline studies provide none of the data that's essential to develop and validate new in silico and in vitro alternative methods. So this is one of the impediments to understanding what the characteristics of these new methods are. Is we're not even generating the data that will allow us to compare new methods with the current method. So let's take a step back and look under the hood in terms of how we actually conduct exposures. So inhalation exposure laboratories really require a significant infrastructure in order to conduct our exposures. And it starts with something as simple as a steady air supply. We have multiple generation systems for generating all classes of test atmospheres that then have to be conditioned and delivered to an exposure chamber, which is designed to provide uniform exposure concentrations to all the animals that are being exposed. The chamber is instrumented with various environmental monitors, and we have to be able to determine what the concentration of test material is within the exposure chamber. And then the material is exhausted, cleaned, and then the cleaned air is then exported outside of our testing laboratory. So whether we use whole body exposure chambers, in which the animals are exposed to the test material within home cages, or we use nose-only exposure systems in which the animals are individually housed in small plexiglass tubes so that only their nose is exposed to the test material, and so they're only breathing in compounds and not being exposed dermally to the test materials. It's a very complicated system, and it requires a lot of overhead in order to conduct these studies. So we do exposures to gases, vapors, and aerosols. So every lab does all these exposures. Vapor studies are often quite difficult to conduct. The vapor is really just the gas phase fraction of a volatile solid or liquid. So the vapors can be really sticky. At high concentrations, you run the risk of moving from a vapor into an aerosol at the transition point of saturated vapor concentration. And during the generation of these vapors, you oftentimes run through a transiently explosive concentration that you have to watch out for and be prepared to eliminate the potential for explosions. So now that I talked about exploding test materials, let's really concentrate on aerosols, which are a lot easier to deal with. Aerosols are characterized not only by the type of test materials, but by the aerodynamic diameter of the population of particles in the test atmosphere. So we have to find out what the aerodynamic diameter of the population of particles is. And the gold standard is really based on gravimetric determination of particle size distributed through a cascade impactor. But because you actually have to weigh the mass of test material collected on each of the stages, low concentration aerosols can be hard to analyze. And so now there are options, including aerosol particle size spectrometers, or APSs, that allow us to infer the particle size based on indirect means, such as time of flight or the reflectance and light scattering characteristics of the aerosol particles. The reason I keep on driving home the importance of aerosol particle size is that for aerosols, the aerodynamic diameter is important because it determines where in the respiratory tract the particles are deposited. So here on this slide, on the left, I have a pictogram of human respiratory tract, and it's color-coded, so it can be broken down into 3 regions. The head region, which is blue, which includes the nasal and pharyngeal and laryngeal tissues, the tracheobronchial or conducting airways, and then the alveolar region, where the gas exchange actually takes place. And then on the right, these graphs depict the size-dependent fractional deposition of particles within each of the regions of the respiratory tract. Each one has a vertical line that is centered on 100 nanometers. And I place that line there because that's the break point for nano-sized aerosols, below which, so anything that's 100 microns or less is effectively classified as a nanomaterial. And in this zone, the deposition is really driven primarily by diffusion, as opposed to other impaction, sedimentation, things like that occur at larger particle sizes. Now, the health effects of inhaled aerosols can be linked to the aerodynamic particle size distribution. So we oftentimes talk about the inhalable fraction of an aerosol, and so that's the fraction of particles that can enter the body through the nose or the mouth during breathing. And essentially, it sets an upper limit to the size of the aerodynamic particle size that's important, because anything above 100 microns really can't be inhaled and deposited anywhere in the respiratory tract. The thoracic fraction then is a smaller subfraction of total inhaled particles, which are less than 30 microns. And so particles in this size range are important for development of asthma, chronic bronchitis, and lung cancer. And then finally, the respirable fraction, which a lot of people are familiar with because this is a size fraction that comes into play for human risk assessment, is defined as the population of particles 10 microns or less. And so really, in terms of human impact, these are associated more with chronic respiratory diseases, pneumoconiosis, and emphysema. Layered on top of the deposition is the whole idea of dose. And so unlike oral or IV or dermal dosing routes, the term dose actually can have several meanings in an inhalation study. So first of all, there's inhaled dose, which is what most people mean when they say dose for an inhalation study. And it's really just the concentration of the test material times the minute ventilation times the duration of the exposure. And so you can calculate then the inhaled dose based on 100% deposition and absorption of the test material. And this is a conservative position, and so this is how we effectively determine dose in most experiments. A better estimate of what actually is the animal is exposed to, or human is exposed to, is a deposited dose, which is that inhaled dose times the fractional deposition. And then finally, what we really want is the absorbed dose, or if you're interested in the bioavailability of the test material. And so that's the deposited dose times an estimate of mass transport or flux into the animal across the epithelium and into the circulation. So I've pointed out that there are no alternative methods currently approved as replacements for acute inhalation tox testing. But as Ian pointed out, there are currently processes where we can waive the need to conduct an acute inhalation study. And Ian provided many more examples, but I wanted to just focus on the three big ones here, which is, first of all, sometimes it's just impossible to generate a concentration high enough to actually result in a toxic insult in these experimental models. And this can be the case for gases, vapors, or aerosols. Now, you can't just say that you can't do it. You have to actually document it, and you have to show what steps that were taken in order to achieve the highest concentration that was available. But if you can demonstrate that, then there's no reason to go ahead and conduct that study. You can also waive a study based on very low volatility, and so there are cutoffs then for both indoor use of products and outdoor use of products. But basically, if the material is not aerosolized, not heated, evaporated, or made inhalable in any way during use, storage, or handling, or it's contained within a non-volatile matrix, then there's no need to conduct the acute inhalation tox testing. And then certain materials, simply based on their physical properties, are too big to be inhaled. And so basically, if the test material greater than 99% of the particles present exceeds 100 micron aerodynamic diameter cutoff, then you can waive that inhalation study. Now, this has to pertain to the base test material. You can't argue that you're spraying something with an aerosol generator that generates aerosol droplets bigger than 100 microns. That's not going to get you a waiver, because there's fragmentation of the particles and there's a potential for an inhalable material. And finally, for these large aerosol particles, you have to also be able to show that they're resistant to mechanical size reduction by attrition. But if you meet those specific requirements, then there's no need to conduct that study. So now I'd like to switch gears a little bit and talk about other potential approaches. First, in silico approaches for replacing or providing additional data for acute inhalation studies. Currently, the utility of in silico predictive models for acute inhalation toxicity is rather limited. And as Amy pointed out, in the future, one of my colleagues, Dan Wilson, will be talking in more detail about these in silico methods. One thing that he'll bring up is that there's a real need for more curated inhalation data. We've spent a lot of time curating the data that's available, but it's only a small fraction of what's held across companies and across jurisdictions. Currently, it requires a significant amount of expert judgment in order to use the tools that are currently available. So you can't just click a few buttons and come up with an answer with models as they currently exist. In testing a commercial QSAR model for acute rat inhalation toxicity, we've demonstrated that it really is very poor. It has very poor sensitivity and gets things wrong often. And so at this point, read across of acute inhalation toxicity to intravenous or oral data is actually better than the global QSAR models that are available. Now, there are several alternative in vitro models that are currently being investigated. And these range from simple submerged cultures, but the question of cell type is still up in the air. Now, will any cell do, or does it need to be a site-specific cell from a specific region of the lung? And can it be a transformed cell, or does it have to be a primary cell? A significant advancement, and where a lot of the research is going now, I may be biased because this is also a system that we're interested in, is the use of air-liquid interface cultures, which plays on the fact that the airway epithelium is unique in that it's constantly exposed to the air-liquid interface. And so these experimental systems are designed to provide that microenvironment for the cells, whether they're transformed or primary human cells, or they're more highly organized 3D organotypic models in which the cells actually differentiate and maintain their normal morphologic characteristics, including the production and secretion of mucus and the activity of ciliated cells at the air-liquid interface. The beauty of these alley cultures is that now we can do the same exposures that we've done in vivo for decades on these same cell types and understand better the intricacies of cell dosimetry and dose to specific cell types that will allow us to make comparisons back to the in vivo model. And then finally, there's a significant amount of work that's being done on organs on a chip, and the lung on a chip has progressed tremendously over the past few years. And it may be possible in the not too distant future to incorporate multi-organs within a testing system with both a lung chip and multi-organ chips linked through microfluidic channels, and which would allow us to investigate systemic effects. But no matter what we do, we have to keep in mind that we have to be able to show equivalent dosimetry in vivo and in vitro. Otherwise, none of this is going to make sense, and we just have to really focus on making certain that we're comparing apples to apples. And keep in mind that we're interested in effects to humans, not comparing the effects of in vitro systems necessarily on rats. So we just laid out what's possible. Why don't we already have these methods? Well, not to be flippant, but it's a lot harder than that looks. There's lots of different cell types in the lung, over 40 different cell types. The cell types change along the length of the respiratory tract. So how do you even know which is the most sensitive target population that you're dealing with? So what cells do you want to use in your in vitro model? The dose of the cells then varies along the respiratory tract too. The physical state of the test material and the location of the respiratory tract then matter in terms of the exposure response characteristics of the system. Acute toxicity may involve point of contact and or systemic effects involving other organ systems. How do you model that in an in vitro system? The mechanism of toxicity varies with the material. There's different types of kinetics by inhalation route. One of the simplest reasons for that is that there's no first-pass hepatic metabolism associated with an acute or with an inhalation exposure. But in fact, there may be local metabolism, which contributes to the toxicity that we see. So, I don't want to, let me summarize this to say that, currently there are no validated and accepted guideline-compliant non-animal testing methods available that meet the needs for hazard assessment for any regulatory authority. But that doesn't mean that there are not alternative methods currently being developed and of great use for non-regulatory needs. So, currently, certainly within our company and others, there's growing acceptance of the use of predictive in silico and in vitro models for hazard assessment. But in order to gain acceptance, the models have to be robust, they have to be transparent in how they operate and where the data is coming from. They have to be reproducible and accurate. And so what we find is that we're getting greater impact and acceptance by introducing these methods early in the development cycle of products, where they can really have an impact in terms of our ability to flag materials of high hazard. Right now, we can incorporate data from alternative methods in the read-across arguments. And so we can anchor the predictive methods to existing animal data. So this helps to build confidence in the models themselves, their predictive power, and it also gives regulatory authorities a chance to see, how the field is progressing and how predictive these alternative methods actually are. We have to be smart when we're implementing the test guidelines. So if you have to test smartly. know what your regulatory need is. Do you need a point estimate, or is a hazard classification good enough for you? And use the existing data that's available. An acute inhalation test is never the first in an acute series, so there's always additional data by other routes and with similar structural molecules generally. And what we know from the past is that simplification and standardization of alternative model systems is going to facilitate regulatory acceptance. So it's great to build your own mousetrap, but that's not what is ultimately going to help facilitate acceptance across regulatory boundaries. So just in summary then, it's really up to us to be the advocates for change and to work smart in order to advance these alternatives to the current paradigm of in vivo inhalation tox testing. Thank you for your time, and willing to take any questions you might have.
Thank you, John. We do have, we're almost out of time, but I'd like if you and Ian are both able to stay on the line for a few minutes for some questions. And thank you to you and Ian. You've really started us off with a great first webinar to our series. If you haven't already, please type your questions in the toolbar on the right-hand side of your screen. And I will hand it over now to Warren Casey, who will be able to ask your questions to the presenters.
Okay, thanks, Amy. And thank you very much, Ian and John. There are a couple of questions. So the EPA, US EPA has recently put out a document for comment where they're proposing to waive the requirement for dermal LD50 testing if you have acute oral LD50 testing for formulations. Do you think an approach like that would work for inhalation, looking at just category to category comparison, not necessarily the dosing, but if something's classified as category 1 or 2 orally, do you have any feel for how those would be classified by inhalation based on your experience?
Well, Warren, this is John. Dan may speak to this when he talks about in silico methods, but what he's seen is that for the most toxic materials, category 1 and category 2, that if you compare oral data with inhalation data, so if you tested the same material by both routes, there's more similarities than, they're more similar than different. And so what you find is that at the higher toxicity classes, oral is a pretty good predictor, although you can be fooled, and so sometimes you will get a material which is more toxic by the inhalation route. And will it be a total replacement? I'm not certain that there would be enough comfort for that, but certainly for the higher concentrate or the higher toxicity classes, it's a pretty good indicator.
Yeah, so that seems to imply that it's really the systemic toxicity that's the problem, not the target organ or the airway. That's
you'll never
maybe there's a.
Yeah,
I was just going to say. Go ahead, John.
Oh yeah, I was just going to point out, and your point is well taken. You'll never point out or pick up the things that are real point of contact toxicants using just the oral data.
May I add to that? Because we did some of the work a while ago using our old NONS database, which is a European regulation, and we showed that the acute dermal and acute oral data, there was a lot of equivalency. If something was very toxic by the oral route, it's likely to be toxic by the dermal route, but things which weren't toxic by the oral route were not toxic by the dermal route. And we didn't have enough information to look at the inhalation route of exposure, but some sort of similar exercise might tell us whether we can start to bear down on the absolute requirement for acute inhalation testing. And I think as John picked up on, that the thing that we noticed with the dermal data was the things that were showing more toxic by the dermal route was because of local site of contact, as in they were corrosives. And it might be that we could weed out the corrosives similarly.
Yep. One more question that you both touched on, and that's the newer technologies, the tissue chips, and any other technologies. Do you see any way around having to compare the performance of those with animal results? I can see a regulator being willing to accept the more restrictive classification based on a newer technology. In other words, it's more restrictive than the animal, but I think it's going to be extremely difficult to convince the regulator that if your in vitro method is safe and it's highly toxic in an animal, that they're going to buy into that without a lot of comparisons being done to the animal data. Do you see any way around that?
The short answer is probably no, but I think a lot of that comparison work, it should be done in the validation stage, so that it gives us a lot of confidence that what the in vitro model is telling us has some sort of in vivo equivalent. And I think that's what I was trying to hint at in my slide, really, that we don't necessarily need LC50 data, but we need to be given as regulators something that we can work with. I think the organs on a chip may well give us some information on whether the substance is toxic to the lung, toxic to the upper respiratory tract. I think, as John was hinting at, that perhaps the more interesting development would be lining up the organs on a chip. So instead of having one organ on a chip, you got rodents on a chip or a human on a chip, and you can link the organs together, because that's one of the things that we wouldn't necessarily get, is information on target organ toxicity that wasn't the lung. That's something that would be really, really useful. I think equally information on respiratory tract irritation, because we don't often get useful information on that from current models, current in vivo models. So the in vitro models might actually give us value added. Then whether John wants to add to that.
No, I really agree. And unfortunately, Warren, I agree with you that early on, at least, there's going to be some resistance to simply believing in the in vitro models. And so it may be a multi-step approach where we're using not only human in vitro models, but also models that still utilize rodent cells or precision cut tissue slice in order to establish the equivalence of response. And as Ian just pointed out, some of the non-lethal endpoints may actually be points where the in vitro models can really shine. So for instance, with the air-liquid interface culture, she has the ability to look at changes in ciliary beat frequency, which is a indirect indication of irritancy that can be explored, or changes in epithelial resistance, which doesn't necessarily correlate with cell death, but rather changes in permeability across the membrane. So we need to not just think about making certain that these are predictive of LC50, but actually provide a hazard assessment, maybe based on non-traditional endpoints.
From the question panel, there's one, can you better define evident toxicity? Can you provide an example for the type of effects which could be termed evident toxicity? What protocol is used to ensure that a definition of evident toxicity is objective?
Well, it's not my work, but it's part of the team that did it. It was led by the UK's National Centre for the 3 Rs, and they spent a lot, 2 or 3 years, pulling together what clinical signs of toxicity would appear to be predictive of severe toxicity or lethality at the top dose. We've published a paper which pulls this all together and defines what we regard as evident toxicity. We've developed, if I remember rightly, 3 criteria, one of which is a 10% decrement in body weight. I think it's hypoactivity I can't remember what the other one is off the top of my head, but I can actually give you a link to the publication which pulls this all together. If that would be really, I can send that to you, Amy, and you can
Yep.
put it up.
Yep, that would be great. I can put that in the follow-up. Another question is maybe for John. Can you comment more on the need for curated data? And it's a little bit of a long question. If we're indeed moving away from in vivo inhalation testing, the data from these legacy studies become increasingly valuable. Any thoughts on creating incentives to make these data available and on creating standards for communicating these data? PDFs of data tables are of limited use, and detailed protocols on how the test was performed and data were collected.
Yeah, you sort of summarized all the really hard parts about collecting and curating that data. There's a lot more important information than simply the concentration that was used or identified as the LC50 in these studies. Time to death is important, the type of test material, a really good understanding of whether it's a liquid or an aerosol or a gas or a vapor. Even when something is called a vapor, you really have to double check and make sure what the concentration was, and then double check to see if it's even possible to have a vapor at that concentration. We've seen cases where the test materials or the atmosphere has just been mischaracterized. As far as collecting and disseminating the data, I mean, it almost seems that there needs to be sort of an independent third party that can sort of be the clearinghouse for data from all sorts of businesses, because I'm certain that some of this data is considered business critical, and so if there's a way to de-identify some of the test material or where it's associated from and incorporate both data from pharma as well as the chemical industry, so the more data, the more correlations we can make, the stronger the predictive models, I think, will be.
There's a similar question then about data curation. It says, are you thinking about applying evidence-based methods and a version of systematic review tool for data curation? This could help in getting quality data on inhalation toxicity.
I have to say that that is way outside of my skill set to answer that question.
Yes,
Right.
I think likewise.
Okay. Let's see, we have one here for you, John. It says, could you elaborate on read across from oral to inhalation, which you showed on slide 16? Could this be applied to mixtures versus single chemicals?
I don't want to come across as being a real advocate at this point in time for using the available oral data as a substitute for inhalation exposures. What I am really comfortable in doing is taking advantage of the oral data that's available, whether it's a single active ingredient or a formulation containing multiple active ingredients, and to use the available data on those the toxic components in order to predict where I need to be in my testing scheme in order to limit the number of exposures that I need. So can I identify, what we will often do is look through a formulation, identify what the most toxic component is, and then look at the relative concentration of it in a test formulation that we need to test, and then use that oral data as our guideline. And so that's where my diatribe on internal dose comes into play, because what we do is we calculate what the effective inhaled dose needs to be and what our exposure concentration would have to be in order to achieve that. So these are we won't use the oral data as a substitute for our acute inhalation data, but we sure will use it to help inform our selection of what an appropriate starting point is and whether it can be used as a portion of an argument to waive a study. So that gets back to the point where if a material is simply not going to be toxic at a limit concentration based on the available data of the actives, then we can use that as a component of a waiver argument.
Okay, and we're about at the end here, but I want to ask one last question, which is there a mechanism to do pulmonary absorption to waive out of inhalation studies?
Well, the air-liquid interface cultures give you the opportunity to look at transport across the epithelium. But you have to sort of match the target site that you're interested in. So there are models that both represent the conducting airway, which are composed of a mucociliary respiratory epithelium with or without underlying, say, fibroblast components in the system. And so you can use that as a test bed to look at transport, passive transport across the epithelium, measuring changes in the applied concentration on the apical surface versus what collects in the basolateral surface. Other than that, that would be my best suggestion. The gold standard for that is the isolated perfused lung, where you can reduce the number of animals that might be used, but it's still an animal use issue.
Okay, are there any final comments from John or Ian or Warren before we wrap up today?
No, thank you.
None from me, just thanks to the speakers.
Okay,
Well,
okay,
thank
great.
you very much.
Okay, thank you everyone for joining, and we look forward to meeting again next month, where the topic will be in vitro models and dosimetry. Enjoy the rest of your day. Bye.
Thank you. Bye. Goodbye.